class of biology that studies the chemistry of living organisms. Uh, we are going to divide this up into a discussion of carbohydrates, lipids, proteins, and nucleic acids. Okay. Now, some things that might be included in that will include vitamins, enzymes, hormones, and those are all just different types of the first four classes of the macromolecules, and is all incorporated into biochemistry. Humans can ingest all the different types of macromolecules, carbohydrates, fats, nucleic acids, and proteins, and are going to get different amounts of energy from those different molecules. Those nutrients become the building blocks for other macromolecules and us in general. Okay, so again, the four classes of macromolecules: carbohydrates, lipids, nucleic acids, and proteins. And one thing that you can think about for all types of these biological molecules is they all have to deal with carbon at some point. So our first class we're going to discuss is the carbohydrate. And one very simple way of telling if you have a carbohydrate is looking at the molecular formula of the compound. Okay, so when the carbon atoms, hydrogen atoms, and oxygen atoms are in a 1 to 2 to 1 ratio, then you would have a carbohydrate. That means for every one carbon atom, you'll have 2 atoms of hydrogen plus 1 of oxygen. And that could be C6, H12, O6, almost any way of arranging the atoms such that they are in that ratio will make them a carbohydrate. Some of the very common things are glucose, fructose or fruit sugar, sucrose, which is table sugar, lactose, the sugar in milk, starch, cellulose, and glycogen. Smallest and simple carbohydrate, which is sort of the go-to monosaccharide, is glucose. So there's the example of C6, H12, O6. And glucose, again, is sort of our go-to carbohydrate. It's the first one that one tends to have to deal with. So blood sugars are actually measuring the amount of glucose in your blood. Okay. Now, if you have a disaccharide, that literally means 2, or in other words, you have 2 of those molecules, 2 of those monosaccharides together. And so sucrose, table sugar, is a very good example of that. So here's an example of sucrose, where you can take a glucose molecule and a fructose molecule, and removing some water, you'll get a disaccharide of glucose, of sucrose. Now, when you have more than just 2 rings, you can have polysaccharides, and polysaccharides have a large number of the monosaccharides, and starch is really your main go-to for that. Okay, um, the starch. Um, so, for example, the starch in a potato is quite a bit of glucose. Okay, it's just a whole bunch of glucoses all strung together. And so when you break down starch, you get a whole lot of sugar in your system. Uh, Polysaccharides, like I was just saying, like starch, have a large number of the same repeating chain. So you can see on the bottom, okay, the polysaccharide of starch is a bunch of glucose molecules that are just repeated together. The next class of biomolecules are lipids. And the easiest way to remember lipids is that these are fats. Okay, and so since oil and water don't mix, these are things that don't dissolve in water. We have a few places within the cell where these lipids are very important, but the main one we think about is phospholipids or the cell wall. The saturated fatty acids mean that they have one single bond between each of their carbon atoms, and those are all solids at room temperature. When you have a monounsaturated, means you have one double bond in the carbon chain. So for example, olive oil, peanut oil are liquids, and they will all be monounsaturated. We can have polyunsaturated, where you have two or more double bonds, and things like soybean oil, safflower oil, and corn oil are all examples of this. So here's some pictures. So you see at the top, here's a unsaturated, and so with between each of the carbons, you see just a single bond. Now, if you are looking at a monounsaturated, that means between these two carbons, we have a double bond. And if you're polyunsaturated, now we have two or more double bonds between the carbon atoms. One of the things, and sort of the reason why this is important, that we know the saturations of the oils, is the shape that the molecule is going to have. So in a monounsaturated, because you just have that one double bond, you get a basically a very bent molecule, and you have one bend in that molecule. In a polyunsaturated, now we're going to have two bends, and in this case, it almost straightens it out, that we have one bond going one way, one bond going the other way, one double bond going one way, one double bond going the other way, so that we get a polyunsaturated. Now, where that becomes really important is in our biomembranes. And when you have a biomembrane, you have a dual layer or phospholipid bilayer. And so you'll have one side near the outside of the cell in the liquid, one side on the inside, with the liquid, and then you have the carbon chains on the inside in the sort of space that's formed between the two sets of polysaccharides. And when there are lots of these bent molecules, this is what's going to keep that fluid, because the molecules just can't get close enough to form a solid. Now, if we're on the right panel, again, now we have very straight, or mostly straight, hydrocarbon chains, and those are able to stack together much better, and that's why you're going to be able to have very very dense and very solid fats. There are the proteins, and proteins, I think everyone has sort of a feel for what proteins are, but they are one of the most essential of all the chemicals inside of a living cell. We can have things, anything from structural things, or they are the enzymes that allow a cell to work. Okay. One of the most important things is that proteins are polymers of amino acids. Okay. All proteins are going to be able to contain carbon, hydrogen, oxygen, nitrogen, and in a few rare examples, some sulfur. And here are just some examples of different protein functions. We can have structural or enzymatic properties. We do have some storage proteins. A big category which we will learn later in the semester is our transport proteins. So whether you want to be able to transport oxygen with something like hemoglobin, or just pass something across the cell membrane, you'll need a transport protein. So proteins are all combinations of amino acids. And you can see on the bottom of the screen there the generic structure of an amino acid. So you have a carbon in the very center. Then on the amino side, you have a nitrogen with two hydrogens. And on the carboxyl acid side, you have a carbon double bonded to an oxygen and an OH. Now in this picture, where you see that R basically means it's any chain that can be there, okay? And that could go from something very simple, like just a hydrogen atom, to something with a whole lot of carbon atoms attached to it. Now, there are three levels of the protein structure that are in every type of protein. Some also have a fourth level. So the first level is the linear sequence of the amino acids, and this is called the primary protein structure. Once you fold up that primary structure, then you get the secondary structures. And the secondary structures, the way I like to think of them is local folds, so that those are little folds of a few amino acids. Now, when we take those secondary structures and build them up into a three-dimensional chain, then we have the tertiary structure. So the final fold of any single protein unit is the tertiary structure. Now, if you have two chains that have to come together, that would be a quaternary structure. And so you've already made the combination of the 3D structure, and then you're going to build two of those 3D structures together. So here's an example of the different kinds of structures. So on the very top are the codes for the different amino acids and in their linear arrangement. Now in the secondary structure, we're now starting to see some coiling or turning of the primary structure. In the tertiary structure, we're going to get a three-dimensional folded protein, and the quaternary structure, now we have multiple subunits of that tertiary structure all held together. An important part about proteins is that their order can be denatured, or in other words, it can be the structure can be taken off with very simple techniques, say heating. So on the left you see the normal protein, and when you denature it, so just adding some heat, you can get that tertiary structure to go back to its linear or primary structure. There are some cases where the denatured protein can renature. In general, that's a fairly hard thing to do, and one might think of an egg. Okay, so the majority of the protein of the whites of an egg are albumin, and when you add heat to it and you fry an egg, you get denatured proteins. When the proteins denature, they go from that very clear state to the white that you see in the frying pan. Now, just because you take the heat away doesn't make the egg renature, and you know that because the egg whites don't turn back to clear just because you take it off the stove. Of biomolecules are the nucleic acids, and the nucleic acids are fun and are almost exclusively DNA and RNA. And I think all of you know that all cell types have DNA and RNA, but a virus doesn't have to have it. It will either have either DNA or RNA. DNA is your major storage molecule that contains all the genes that will eventually code for all the proteins that we're going to see. The RNA molecule is a molecule for transferring information from the DNA to the ribosome so that we can make proteins from it. The building block of a nucleic acid is called a nucleotide, and they are a bit more complex than amino acids. So we can either have DNA or deoxyribonucleotides, or in RNA we can have ribonucleotides or RNA nucleotides. Any given nucleotide is going to have a sugar, the base, and a phosphate group. Now, hopefully, this is a bit of a review material, but that is that with your bases, an A is always going to pair with a T, and a G is always going to pair with a C. And so those are the AT, CG are base pairs, and to take those base pairs apart, with an AT base pair, you have to break 2 hydrogen bonds, and with the CG, you need to break 3 hydrogen bonds. Now, hydrogen bonds are quite weak, but when you start getting millions of them, it can add up. Okay, and so we can generally denature or take 2 strands of a DNA apart by just adding some heat and maybe heating it up to maybe let's say 99 degrees if it's an average of AT and CG base pairs. So here's a cartoon with DNA, and you can see that there are 2 strands, and because the 2 strands circle around each other, this is known as a double helix. At the very bottom of that figure, you can see the AT and CG base pairs with the number of hydrogen bonds that are between the 2 of them.